Good afternoon, everyone. Thank you for joining us for today's program. My name is Ken Shopman. I'm the Executive Director for ABRF. We appreciate your support, and we certainly appreciate 10X presenting with us today. Before we get started, we're going to just make a couple of housekeeping announcements and other updates from ABRF. And so today's presentation by Egon Regani with 10X will discuss single cell and spatial analysis technologies for genomics cores. Feel free to introduce yourself in chat. That's the way we'll be taking questions. If you'd like to say where you're based, you know, let's see how many countries we have represented in the audience today and how many different institutions. Egon will take questions. Feel free to put them in the chat at any time, and he'll address those at the end. As you heard when you logged in, today's session is being recorded. We will send out the link to the recording to all registrants following the program, and it'll also be posted on the ABRF website. Just a couple of announcements and updates from ABRF. Our brand new compensation survey that's still in place, it closes today, in fact. So perhaps you've seen a link, or some of your colleagues have seen a link. This is a brand new effort, and we think it's very important to try to benchmark compensation across the cores, across regions, and size of institution. So you have just a few more hours today to complete that survey. You can see the QR code here. If you've got a moment, you can scan that. All contacts should have also received a link to the survey as well. For ABRF members, this is the time to vote for the new executive board members. You see the link here as well as the QR code. The election closes at the end of the day on Friday. So you have just a couple more days to cast your ballot for our new board members. And we're launching a very quick one-question survey to understand what other societies you're a part of. Because the degree to which there's overlap between ABRF and other organizations provides us an opportunity to start conversations with those groups about potential collaboration. Joint programs, reciprocal programs, hosting each other's events, and so forth. So when you can, take a moment, you'll see the link, and again, another QR code just to fill out one question on that quick survey. And today, it's membership renewal season. All individual members received their renewal notice in the last couple of weeks. So please take a few moments, renew your membership by the end of the year, just to avoid any glitches, any delays in your access to ABRF resources online. And for those of you that are considering coming to the annual meeting next year, you can bundle your membership renewal with registration for the meeting and save a little bit of money as well. Next week, the ABRF Education Committee is pleased to present this program, The Future Forward in Flow Cytometry, which will be Tuesday, December 6th from 11 a.m. to 1 p.m. Eastern. It's a 2-hour workshop featuring the panel that you see on the screen here. This will be hosted by Nicole White from the ABRF Education Committee. Once again, you can find this on the ABRF website, or if you have a moment, scan the QR code to register. This, of course, is a complimentary program like all the ABRF education workshops are. And finally, we just want to remind everyone that registration is open for next year's annual meeting, which will be May 7 to 10 at the Sheraton, Boston. We're looking forward to a terrifically exciting meeting. It's just a unique venue and unique location for ABRF. I hope you saw the announcement in this week's newsletter of some of the primary speakers that'll be featured, including the ABRF award recipients and our keynote speakers. You can find more information on the meeting website. All right, so I'm going to stop there. And Egon, thank you very much for joining us. We really appreciate it. And I will turn it over to you and take it away.
Thank you, Ken. Thanks a lot, and thanks for the introduction. Let me just go here. Full screen, hopefully you can see my slide and my pointer. Yep.
Yes.
Thanks, Ken. Thanks for giving us the opportunity to present here today. And thank you everyone for joining us today. My name is Egon Rangini. I'm one of the science and technology advisors for 10XGenomics. And today I'd like to provide you with an overview of some of our new solutions that you can use now to also to profile fixed samples along with fresh samples. So before we start, I'd like to ask you if you have a chance to scan this QR code, so we can get your information, can then reach out to us. And also I'd like to quickly introduce my colleague, Adriana Suarez. Also, she's a science and technology advisor. She'll be helping with the question. So as Ken mentioned, feel free to ask to enter your question in the chat button, and then we'll try to answer them as we go through the presentation, and then at the end we should have some time for Q&A. So, and one more thing before I start, you can have this presentation. I'll be more than happy to share the deck. Now, what I like to start with is this slide here, which shows the 3 platforms that we've been developing at 10X to puzzle the complexity of biology. Today, I'm going to start with the Chromium platform. That is what includes everything for single-cell genomics. I'm going to then switch to the Xenium platform for spatial transcriptomics, and then I have some slide about our newest platform, which is Xenium, and this is an instrument for in situ analysis. Now, when we look at our technology, I would say we are really proud to see that our technologies have been used in more than 4,000 publications, and it's very exciting to witness what researchers have been able to achieve by using 10X technologies, and generally speaking, how to advance the understanding of biology and not only the research field, but the understanding of biology in general. And so talking about the single cell platform, so this is a Chromium platform. This has been already featured in top tier journals and included multiple times in the top 10 innovation list by scientists. Now, when we look at the workflow, with 10X, essentially we look at what we call a beginning to end workflow, in which all reagents that you need to make a library, those are included in the kit we provide. And so when an investigator wants to run a single cell experiment, what they would need to prepare is a single cell or single nucleus suspension. And then typically this is the input that is submitted to a genomics core, for instance, or a core where an instrument will sit. The core will partition the cells or nuclei, then they will make a library, and oftentimes they will also sequence those libraries. And then in this workflow, we also take care of the opposite side of the workflow. That is where we provide fully supported, fully integrated bioinformatics tools, bioinformatics pipeline for data analysis and data visualization. And those tools comes at no cost for you, including our cloud analysis. That it's a fairly new solution that allows you to run a Cell Ranger, our single cell pipeline, for free using our cloud. Now, the majority of those 4,000 plus publications have been generated using human and mouse samples, but there is a really good number, an increasing number, I should say, of publications generated using what I would call non-traditional model organisms. So that can be rats, non-human primates, it can be plants, fishes, insects, and so on and so forth. And when it comes to sample type compatibility, here is really where we have solution approaches, recommendation that allows us to start for pretty much a lot of different type of tissues that would include, or samples, I should say, that would include single cell, single nucleus suspension. We can profile fresh tissue, frozen tissue. We can now also fix tissue, something I'm going to talk about today. And we can also now isolate single cell from FFP tissue blocks. That's a new protocol, and we are super excited about that. Now, just want to show you one example of how these technologies have been used in the field. And I chose this 2022 paper in Nature Neuroscience. This publication came up at the end of August, beginning of September. So it's very recent. And the authors here use the single cell gene expression solution to profile more than 430,000 nuclei isolated from brain samples from patients that had frontotemporal dementia, and also control, of course. And essentially, by looking at the differential gene expression, looking at different cell population, they identified a population of astrocytes, which is this one in particular, gray matter astrocytes, which seems to be associated with the blood-brain barrier dysfunction. And so this is a great example that shows you really how you can use a single-cell approach to really unpuzzle or look at into the heterogeneity of a very complex sample, such as those brain samples. But we know that biology is extremely complex, and if we really want to understand the full complexity of biology, what we want to do is to capture or measure multiple types of information simultaneously from the same cell. And this is what we call multiomic cytometry. And so we have now tools and solutions that allows us to simultaneously measure gene expression and expression level of cell surface protein, that is by labeling cells with oligoconjugated antibodies. We can look at CRISPR-mediated gene perturbation. We have a fantastic tool to study the adaptive immune system. With that, we can measure gene expression and profile the full-length VDJ sequences of the T-cell receptor or the B-cell receptor. We can also look at protein expression and CRISPR-mediated gene perturbation. And one example, actually, of these multi-omic approaches is this paper here. It's a 2021 paper. The goal here was to understand why oftentimes COVID symptoms are milder in kids compared to adults. And so to address this question, the authors profiled more than 700,000 cells from pediatric and adult COVID patients, and they generated a truly multi-omic data set because they generated gene expression data libraries. They generated T-cell receptor and B-cell receptor libraries, so VDJ libraries, and they also generated cell surface protein libraries. This is CytSeq. And by combining this really wealth of information, the authors were able to demonstrate that the adaptive immune system of children shows a significantly higher clonotype diversity compared to adult. And essentially this might explain, at least in part, why sometimes kids, when they develop COVID, the COVID symptoms are milder compared to that adult symptoms. And at 10X, we keep innovating. We keep on innovating. And so recently, we launched a new new solution called barcode-enabled antigen mapping, or BEAM. And this is a solution that will allow you to interrogate VDJ receptor or antigen-specific, I should say, VDJ receptor in T cells or B cells with single-cell resolution. And so the investigators here will be able to profile gene expression and then simultaneously match the individual receptor sequence with the antigen specificity, with the antigen information, again at the single cell level. And to give you an idea of how we use this solution in-house, here we profiled 4.5 million B cells from a COVID patient. Those were isolated from a blood sample. We stained those cells with a panel of antigens specific for 2 of the spike proteins of the SARS-CoV-2 virus. And then we fact sorted the cells, we generated libraries, and overall, we identified thousands of high-quality B-cell receptors that could recognize those antigens. We looked at 239 of those BCR sequences for the study. And then we used the information from BEAM to essentially express those antibodies. We recovered more than 200 antibodies that could bind the spike proteins with very good affinity. More than 50 of those antibodies had a IC50, which was lower than 200 nanogram per ml, and which essentially will place them pretty much in the same range of potency shown by other commercially available neutralizing antibodies. So this will really be a powerful tool to identify, for instance, neutralizing antibodies, and now it's available for you can order it. And again, in a very 10X fashion, we are also taking care of data analysis. And so there's going to be a new version of the Loop VDJ browser, which now can be used not only to identify different clonotypes, and so each group of cells here, each group of dots here represent the clonotypes, but now we can also use this version of Loop to essentially clonotypes will be color coded based on their antigen specificity. And so investigator will really be able now to associate a particular clonotype with each antigen that they are screening. And those applications, those solutions can be run on our Chromium X, which is the I'll say latest and greatest instrument, is what we consider being the future for single-cell genomics. That's because it's really is the most flexible instrument that we have released so far. And it's a system that allows us to scale up the size of our experiment. We can and allows us to essentially profile up to 1 million cells per run with a significant reduction in cost per cell. The cost can go down to 75%. That is one instrument we have at 10X. We also have a fully automated platform, which is called the Chromium Connect. And this is an instrument that allows investigator cores to generate 3' or 5' libraries starting from a single cell suspension. And so the Connect here in about 8 to 9 hours, I should say. And so the Connect is really intended to generate highly reproducible, highly consistent data, as we can see here, when compared to the manual workflow, and really optimize the lab personnel time and the lab resources in general. Now, I would like to go back to this slide here and talk about a solution that we developed to interrogate gene expression and chromatin landscape from the same cell. And this is what we call the Multiom kit, or Multiom ATAC plus gene expression, Multiom for brevity. And so for the first time with this kit, we have a unique opportunity to really interrogate the interplay that occurs between epigenetic regulation and gene expression. And this is really critical if we want to understand how, for instance, cell lineages are established or how cells simply respond to a particular stimulus. And so with the Multiom kit, we start from a single nuclei suspension. So here we prepare nuclei. We use this single nuclear suspension to generate 2 libraries, a gene expression libraries and a single cell ATAC libraries. And those 2 libraries can be directly linked together because they share the same pool of 10x barcode. And so, for instance, we can think about using, let's say, gene expression markers to look to annotate the gene expression cluster, then simply transfer the annotation to the ATAC space and identify those cell population as well. But the important thing here is now we can really start looking at correlation between a region of open chromatin and gene expression patterns. So we can start really to understand how region of accessible chromatins will impact gene expression in a positive manner or in a negative manner. Now, for this solution, as I mentioned, we need nuclei. And we knew that isolating nuclei from tissue can be challenging. And so recently, we launched a nuclei isolation kit, which is compatible with a large variety of tissues. All you need is 3 to 50 milligrams of tissue, a benchtop centrifuge, and 1 hour of your time. The entire nuclei isolation occurs in an hour. And at the very end of the workflow, you end up with a high quality single nuclei suspension that requires no optimization or very little optimization. And in a very 10x fashion, everything you need for nuclei isolation, including the RNase inhibitor that is part of the kit we provide. And so to show you how we use this kit internally, we extracted nuclei from a frozen human kidney tumor samples, and we generated multiom data, apologies. And so we were able to recover immune cells, we were able to recover kidney specific cells, in particular those podocytes. And I'm a kidney guy, and so this is really nice to see that we were able to retain so many podocytes. And for you who are not very familiar with kidney, so podocytes in human, especially in human, are very large cells and they present many processes. And so sometimes it might be tricky to capture them, but here, thanks to the nuclei approach, then we can retain a very high number of those podocytes and have a beautiful representation of the data. Now, what I showed you so far are solutions that require using living tissue or living cells, living nuclei. We know that using or working with living tissue can be challenging, especially from a core perspective. We know that you might have clients who have sample available outside their regular business hours. It can be during the weekend. We also know that there are investigators who might want to collect samples over multiple time points. Let's say they are running a longitudinal studies. And so it would be better to be able to collect those samples, store them, and then perhaps send those samples to their core in a batch. We also know that samples would degrade. That's a huge challenge where you perhaps get samples from customers, and we need to really make sure that viability stays as high as possible so that we can generate high quality data. And so stabilizing samples is something really critical, not only for large or complex project as is shown here in this slide, but I would say for any type of project, any type of number of samples. And then there can be also logistical constraints in which, again, you might be receiving samples from a client that is not really in your building, or you might be sending samples to someone else, to a collaborator. And so to essentially address all those challenges, very recently, we released a new solution, which initially was called the Fixed RNA Profiling Kit. Now it's called Single Cell Gene Expression Flex. But it's essentially is a solution that allows us to fix our samples in 4% formaldehyde, and then store the single cell suspension or the single nucleus suspension for up to 6 months at minus 80 degrees. It was 3 months. Now we just extended it to 6 months. And so with this solution, we can now really collect samples whenever they are available, whenever they become available, and preserve the biological state of the samples, because samples will be fixed. And with this solution, we use a probe-based chemistry in which set of probe pairs has been designed to account for all annotated isoform of a particular gene. We have more than 55,000 probe pairs that will recognize or will target more than 18,000 coding genes in human or in mouse. So probes are for human or mouse. And one thing I like to mention here about the mRNA target size. So this sequence here, the sequence that is recognized by the probes, this is 50 nucleotides. And so even when the RNA is, let's say, partially degraded because of the fixation procedure, that's normal. But we can still detect those transcripts because of this probe-based chemistry. Thanks to the probe-based chemistry, we actually see a significant increase in sensitivity when we use this kit compared to the typical 3' chemistry or the typical 5' chemistry, I should also say. And we were also able to reduce the minimum recommended sequencing depth to just 10,000 read pairs per cell. So, you know, your clients, you'll also be able to save money on the sequence when sequencing those libraries. And what I'm showing you here are not cherry-picked data. So this is a trend that we see across multiple samples. And now it has also been essentially verified or confirmed, I should say, in the field. This is a manuscript from Luciano Martellotto and Jasmine Plummer, and they use the fixed RNA, or I should say the Flex kit now, and they compare it against the 3' chemistry on the same sample. And they show that the Flex chemistry could detect more than twice the number of transcripts compared to the 3', and crucially, it could detect even more, could better detect lowly expressed genes, including transcription factor. And what you see here are the top 19 or top 20 transcription factors they detected in this assay. And also, I'm very excited to show you some very new data. So this is a comparison that was run by a lab, which is not affiliated with us, so it's not affiliated with 10X. And in this experiment, they compare our Flex kit against the PARS EverCode V2 chemistry on the same sample. It was human PBMCs. And again, they found that our chemistry is significantly more sensitive compared to PARS. And so with Flex, really, you get an unmatched sensitivity, you can detect more genes, you get more data, so a better understanding of biology, and that is achieved by also with a lower sequencing depth. Now, we also wanted to make sure that you can store cells or samples after fixation, and you still have the correct representation of cell population. And this is exactly what we did here with PBMCs. So we store the cells We fix the cells and we store them for up to 6 months at -80. And as you can see here, the proportion of different cell population were not affected by the temperature or the length of time that we tested. Now, with the something also I will mention later, but with the fixed RNA or the flex kit, you can fix single cell, single nuclear suspension, but you can also fix tissue. So you can take a tissue that can be fresh or snap frozen. You will need to chop it or mince it, then you fix it. That tissue can be stored at 4 degrees for up to 1 week, and then you need to isolate single cell. And those cells, again, can be stored at -80 for 3 months because they have been fixed. And so in this particular experiment in-house, we tested a human endometrium sample that was, so we use a fresh tissue and a snap frozen tissue. The 0 day here means that we fix the tissue and then immediately isolate single cells. 5 days, 4 degrees means that the tissue was fixed, then was stored at 4 degrees for 5 days, and then we isolated single cells. And as you can see here, there is really minimal differences in terms of cell population detected across those conditions that we tested. Now, this is also a multiomic solution. That's because we can simultaneously measure gene expression and profile the expression level of cell surface protein by labeling cell with oligoconjugated antibodies from BioLegend. That will be the total CPB antibodies. Just keep in mind that the cells would need to be labeled before fixing them, and sample will need to be run in a single plex format, not in a multiplex format. Now, talking about multiplexing, that is the other great, or one of the other great advantage of this chemistry. We can now multiplex samples together and so run them in the same channel of the chip without labeling cells with oligoconjugated antibodies or oligoconjugated lipid molecules. And that's because we included a unique barcode sequence to each set of probes. And by using the information of these barcodes, we can demultiplex samples during data analysis. So if you think about that, now the advantages are, well, we don't need to buy antibodies anymore, and we don't need to decide up front which sample we want to pull together. With Flex, we simply pull together samples the way that makes most sense for the experiment when we start the single cell workflow, not when we collect the sample. So an investigator will technically have up to 6 months to think about how to combine the samples if they want to do that. So we can combine up to 16 samples together in the same channel and target up to 128,000 cells per channel. If we were to run a full chip, that would be 1,024,000 cells per run. And of course, when you multiplex samples, there is a cost-saving factor that is associated. So the more sample you multiplex, the less the cost per sample would be. Now, the other great advantage for this kit is, of course, the flexibility. Now, probes that we have are compatible for human and mouse. But when it comes to the type of sample that we can fix, here is really where we can fix a lot of different sample types. And we have protocols to do that. So we can fix cell lines or cells, I should say, nuclei. We can fix fresh tissue, snap frozen tissue. Remember, you need to chop it first and then throw in the fixative. And then we also have this protocol that allows us to isolate single cells from FFP tissue blocks. And this is what I'd like to show you here now. So in a nutshell, with this protocol, you will be cutting a couple of curves or scrolls of your FFP block, dissociate the tissue, isolate a single cells, and then profile the single cell with the Flex chemistry. And this chemistry rather, has already been proven successful in the field. And so this is actually the bioRxiv paper that I mentioned earlier. It was the one published by Luciano Martellotto and Jasmine Plummer. In the first part of the study, they did that comparison between Flex and 3'. In the second part of this study, they developed a method to isolate nuclei. But before going there, I just want to point out to Luciano's tweet here. I really love it, where he said that the cure for cancer is sitting in a pathologist drawer. And so what they did in the second part of the study was to develop a protocol to isolate intact nuclei from FFP blocks. So it's pretty much similar to what we do with our protocol. We isolate cells, Luciano's group isolate nuclei, and they call this method SM-patho-seq, so single nucleate patho-seq. And so in this paper, they compared the performance, or they profiled those nuclei with the flex chemistry, and they compared the data against a matched FFP sample that was profiled using Visium for FFP. And they showed a significantly high correlation in terms of gene expression, in terms of the type of cells that were detected between those two technologies. And so in my last bullet point here, I'm kind of paraphrasing Luciano, where he said that now, thanks to this protocol or our protocol for cell isolation, now we can really think about generating high quality and high sensitive single cell or single nuclei data from our FFP tissue sample, which will allow us to perform extensive retrospective studies on our archived sample. And this is really exciting because for the first time now we have a solution, we have an opportunity to generate single cell or single nuclei data and spatial transcriptomics data starting from the very same tissue block, which essentially will give us an unprecedented view of biology. And so to sum up this part of the talk, I'm hoping, or at least regarding Flex, I'm hoping I was able to convince you that now the Flex chemistry, the Flex solution is now the new gold standard for single cell RNA sequencing. And that's because of its really great or superior sensitivity, I should say. It's also an extremely highly flexible kit. We've seen that it's compatible with many different types of samples that we can profile. It allows investigators to run anything between 1 sample and 128 samples per run, or capture up to 1 million cells per run. With Flex, investigators will be able to collect samples whenever they become available again. We'll be able to fix them, store them, batch them, and then submit the samples to the core without worrying anymore about sample viability, sample degradation. That's because, again, the sample will be fixed, and so there's not going to be any degradation anymore, which is going to be great. Now, we also saw that we can multiplex samples. That is a great strategy to, well, profile multiple samples together, reduce potential batch effect due to the different sample collections. And from, I believe, from a core point of view standpoint, it will also help save hands-on time, so you can process a large number of samples in a limited time. And it will also reduce the cost per sample down to 75%. And then last but not least, this chemistry is compatible with FFP blocks, which again is super exciting, I think. Okay, so I'm gonna kind of use the FFP block as a segue to my next part of the talk, which is spatial transcriptomics. Now, what I would say is, with single cell, we generate beautiful data, and we get a lot of insight into the biology of a sample, but we do miss one piece of information, which is the morphological context of where genes are expressed within the tissue. But this is exactly the information we can retain in Visium. So this is our platform for spatial transcriptomics, and so we can interrogate the entire transcriptome of a tissue section while maintaining the morphological context, morphological information of where genes are expressed within the tissue. And again, we are really proud and very excited to see the rapid adoption of Visium. This is a fairly old data. Last time it was updated, it was July 1st. And so definitely now we passed the 300 study milestone. And also we are really proud to see the number of institutions that have adopted Visium, especially when it's compared to other technologies. Now, for Visium, we have 2 chemistries. One chemistry is for fresh frozen tissue sections. That chemistry captures the native mRNA from tissue section. Therefore, it's what we consider being a species agnostic chemistry or solution. With FFP, for FFP tissues, we have a different chemistry. This is a probe-based chemistry, and is, I would say, somehow similar to what I show you for single cell for the single cell flex chemistry. And so we have probes for mouse and human, and those will cover, as I mentioned, the entire transcriptome. The key of the technology is the gene expression slide. And so here is where we have 4 capture areas. Those are the areas where you'll be placing your tissue section, fresh, frozen, or FFP. Each capture area is 6.5 millimeters square, contains approximately 5,000 barcoded spots. Each spot is 55 micron in diameter and is coated with millions of oligos, which terminate with a poly-D tail. And so this is the tail that will capture either the poly-A mRNA from fresh frozen section or the polyadenylated probes from FFP section. And then there is a spatial barcode sequence, which is unique to each barcode, a barcoded spot, I should say. And so this is what provides the address of where genes are expressed within the tissue. And to give you, again, a very quick example is this recent paper published in Immunity. Here, the authors use Visium to characterize the B-cell composition of tertiary lymphoid structures, or TLS. And TLS are aggregates of B cells, T cells, and fibroblasts. And so with this study, the authors had access to 12 renal cell cancer biopsies in FFP blocks and 12 fresh frozen biopsies. So they use the FFP blocks to identify multiple stages of B cell development in the TLS. TLS, by the way, is this region here, so very tiny in this biopsy. And they're also able to identify plasma cells. And then that's very interesting, I think. They use the frozen biopsies and the fresh frozen chemistries to perform a BCR clonotype analysis. And so remember, with the fresh frozen chemistry, we generate full-length cDNA starting from native mRNA. And so the authors here, they were able to identify clonotypes in the TLS, and they were able to essentially demonstrate that TLS show the highest number of unique clonotypes, and they are also the sites where likely hypermutation occurs. And so this is again, I believe, is a beautiful example of how you can use Visium to really interrogate the heterogeneity or complexity of your samples, gives you an idea also of the sensitivity and resolution of this technology. And so we see that the Visium platform essentially makes it possible to combine an histology image with the gene expression from the very same tissue section. But we know that sometimes running the entire Visium workflow might be challenging. And so, for instance, from a histology course standpoint, sometimes those cores might not be fully equipped to run the entire workflow, or they might not be feeling comfortable handling an NGS-based technology assay. Conversely, genomics core might not have a lot of experience with tissue. And so at 10X, we wanted to fill this gap. And to fill this gap, we recently launched a new instrument, which is called the Cytosis. Now, in a nutshell, what the Cytosis does is to allow the investigator to generate gene expression libraries starting from tissue section, FFP tissue sections that have been mounted on regular glass slide. And so it enables the access to even more FFP samples than now we can study. And so we can think about using the Cytosis to study archived tissue sections, or we can think about using the Cytosis to actually choose the best section for our study. That's because we can take, let's say, a section out of a new section out of a FFP blocks. We can place it on a regular glass slide. We can stain it with H&E or with immunofluorescence, and then we can then check whether or not that is really the sample that we want to study, if that is before committing to the entire Visium workflow. And then also with the Cytosis, I'll show you later, we can also profile tissue with different sites. And so the Cytosis here, if we look at the workflow, essentially sits as a bridge between the histology and the molecular biology portions of this workflow. And that's because upstream of the Cytosis, we have sample prep, imaging, and staining imaging and probe hybridizations, and those steps occur on a regular glass slide. Then the slide is placed inside the CytAssist together with a gene expression slide, and the CytAssist will transfer only the polyadenylated probes from the tissue section on the gene expression slide. So that downstream on the CytAssist, what we will be doing is to essentially complete the library prep and then sequence the library. And the CytAssist also comes with an improved version of the Xenium reagents. Thanks to that, we were able to even wider the range of sample input quality that we can now profile. That's because with the CytAssist, we can profile samples with the DV200 greater than 30%. And that is down from, and that is as opposed to the DV200 of 50% that we still recommend with the direct mount option. Direct mount means you place tissue section directly on a gene expression slide and you don't use a CytAssist. Now, as I mentioned, we can profile archived samples with a CytAssist. Even if you have a cover slip on a tissue section, we have a protocol to remove the cover slip. We can think about aligning the target tissue to the Xenium slide area. So for instance, we can decide to look at the head of this embryo only, or at the torso of this embryo, or in between, let's say. The CytAssist also is compatible with a new slide, which features a 11 millimeter square capture area, that is as opposed to the 6.5 millimeter square. And so to give you an idea of how much more tissue you can capture, this is a tissue placed inside a 6.5 millimeter square, and that's the tissue that you will be able to profile in a 11 millimeter square, or that much more tissue that you'll be able to profile in an 11 millimeter square slide. And again, because of the improved reagents and the overall, I would say the CytAssist workflow, it is now also easier to profile challenging tissues. And one tissue that immediately comes to my mind is skin. Okay. So in the last, I believe, 5 minutes, 6 minutes or so, I'd like to introduce our newest instrument, newest platform, I should call it, which is called Xenium. And Xenium is our in situ analysis platform that essentially allows us to interrogate the expression level of hundreds of target genes in FFP or fresh frozen tissue section and achieve subcellular resolution. So this is not an NGS readout, so we don't generate Illumina libraries. It's a microscopy-based readout, and the imaging and data analysis occur on the instrument as the run progresses. Now, if we look at the workflow from a 30,000 feet view, I would say, we start with with sample prep. Here is where we place a FFP or fresh frozen tissue section on a Xenium slide. Then we hybridize our circular DNA probes to the section. Each probe will carry a specific barcode, so we can distinguish different genes. Then the probes will be enzymatically amplified. That is done before loading the slide onto the instrument. And at that point, the tissue will undergo multiple cycles of fluorescent labeling or fluorescent hybridization, I should say, and imaging, which would essentially generate an optical signature for each gene that is detected. And at the very end of the workflow, what we have will be a spatial map of all transcripts detected in or expressed in the tissue. And so to give you an idea of the type of data you can get, this is a mouse brain sample. This is a FFP section. We stain, or we hybridize, I should say, this section with a panel of 248 antibodies. We detected more than 54 million transcripts with a median transcript per cell close to 200. And in total, we detected more than 175,000 cells. And here in this image, think about the different colors will represent different genes. And if we were able to zoom in on the image, we will be able to see individual dots that will represent individual transcript. Those will be inside cells. And so with Xenium, we achieve really true subcellular resolution. And with Xenium, we already have the highest data throughput available right now. when it's compared with other platforms. And we have already built a lot of headrooms in this platform so that we can even increase the throughput in the years to come, even higher than what we have right now. And briefly, I would like also to mention our strategy when it comes to designing panels. So here is where we take into account single-cell data and Visium data that are generated with our platform. Then we look, of course, at scientific literature, and importantly, we use feedback from experts in the field. And that is essentially a strategy to generate a very high quality and very meaningful panels that would not need any optimization, and pretty much they can be plug and play, can be immediately used for studying the samples that you are interested in. And so I will see that as a target approach, really Xenium can allow you to explore the genes you are interested in incredible details. But now we can think about generating even more data from the tissue section that we use for in situ analysis. And so that is, again, thanks to the Cytosis. We can think about, for instance, generating whole transcriptome data starting from the very same tissue section that we use for in situ analysis. So for instance, we got an unexpected readout, and we want at this point to look at instead of hundreds of target genes, and we want to explore the entire transcriptome. Well, we can do that thanks to the Cytosis. That's because the Xenium workflow is not disruptive, and so the morphology and the biological information is retained in the tissue section. And this is just an example of how we can use these two platforms together to study the biology of the sample we are interested in. And in fact, this is just I would say, in fact, we can think about using our entire ecosystem to really start unpuzzling the complexity of biology. This is pretty much what we did in this manuscript that we released at the beginning of October. We studied the same FFP sample. This was a human breast cancer sample, and so we isolated a couple of scrolls. We cut a couple of scrolls and generated single cell data using the flex chemistry. Then we cut a consecutive section and generated spatial transcriptomics data using the Cytosis, and then again a consecutive section that was used with the Xenium analyzer to generate in situ data. And really, this ecosystem is what gives us an unprecedented view of biology. And so to conclude, our goal here is really to provide you with an ecosystem that can really enable you to do incredible research. And we are spending a lot of time, a lot of efforts to make sure that those three platforms can work well together, together really well, I should say, and can also be compatible with as many types of samples as possible. And on top of that, we have outstanding resources at 10X that is to really fully support you, starting from your experimental design, then going through sample prep, all the way down to data analysis. And so what I would like to say here to really conclude is, don't hesitate to reach out to us. We are here to help you. We are here to make sure you're gonna be successful. And with that, thank you for your attention. And I'll be happy to take any question.
Thanks, Egon. This is Adriana Suarez, Science and Technology Advisor for 10X and Egon's colleague. And I have been answering some questions in the chat, but there are a lot of questions coming up. So we have some that we can answer live. The first one is about the Cytosis. So as you mentioned, the Cytosis is our new instrument to facilitate the Visium workflow. And the question was about the throughput and what would make sense for a core. So how many Cytosis instruments do mid and large size cores typically need, or you've seen in the field? How many Cytosis people at cores are acquiring? Is it recommended to buy 2 for redundancy and higher throughput?
Yeah, that's a great question. So the Cytosis has been released, I think it was released 3 months ago, 2-3 months ago. So now we are seeing one placement per core, but yes, it does make a lot of sense to think about having more than one instrument, not just for redundancy, but mainly for a throughput standpoint, I would say if you need to run multiple samples, that would be something that I would strongly consider. Adriana, what do you think about that?
Yeah, I mean, I've seen the same in the field. It really depends on what works and how people want to streamline the runs. The next question is about Visium. So staying on Visium and Visium for fresh frozen, where we Visium for FFPE, where we capture the DV200. So the question is, if we have data on DV200 associated with mean UMIs or number of genes with direct placement with V1, they're curious to know if we have data that indicates that with V1, the DV200 should be more than 50%.
Yeah, that's a good question. So what we recommend is using, if possible, using blocks with a DV200 of 50%. That is what we are seeing in-house, generating robust, reproducible data. What I would say is, it always depends on the particular situation and how precious a sample is, or how many samples of the same type an investigator might have. And so if you check the DV200 of a sample and you see that is 45%, I would say there is a good chance that probably is gonna work. And actually we have seen data in the field where even with a DV200 in below 40%, even in the 30%, it was generating good data. Now, ideally, you want to use a DV200 greater than 50, if that's possible. If that's not possible, again, it really depends. 40% it probably is going to work. If the DV200 is down in the teens, well, that is more risky, right? And so if you have, let's say, only one sample, and that is a very precious sample, and the DV200 doesn't reach 50%, I would probably still think about running it, knowing the potential risk. Again, depending on how low the DV200 is. If you have perhaps multiple samples, let's say 3 blocks, and one block is 25%, the other 2 blocks are 48 and 52, I would probably prefer running the other 2, the 48 and 52, rather than using the block with the lowest DV200. So it really depends on the situation. But again, going back to what I mentioned at the beginning, is in-house we've seen robust data with a DV200 greater than 50. That doesn't mean that if you don't reach 50%, you cannot run the assay.
Yeah, and then I will echo that. One of the main recommendations for researchers is to pilot and to run pilots with a small sample size if they are not sure about the quality, or if it is the first time they run an assay like this. So a pilot can go a long way to help them assess their sample, but also give them ideas on follow-up experiments to make their hypothesis more robust and more targeted. So we can definitely help researchers address those questions. There were also some questions about Xenium in terms of throughput, and we have been going back and forth with the numbers. So I don't know if you want to take these. I was also making the calculations here, so we know that.
I can. So what I can tell is actually, let me go back here. The Xenium, or maybe here, the Xenium slide has something called Imageable Area. And the Imageable Area is 12 millimeter by 24 millimeters. So that means that inside the Imageable Area, you can place a very large tissue section, or 2 tissue sections, or 4 or 8, for instance. It really depends. The instrument here processes 2 slides at a time, and the run takes about 2 days. That means that that means that you have in 1 week 1 week time, you can process up to 6 slides on the instrument. And again, the reason why I didn't say the word number of samples per run or number of samples per week, for instance, is because we essentially think in terms of imageable area. So I guess this slide perhaps explain gives the idea that it's really there is an imageable area. So the number of samples depends on that you can do per week depends on how many tissue you want to place in this imageable area. But when we compare the throughput in terms of, again, millimeter square per week, that is really where Xenium is on a different level compared to other commercial other platforms.
Yeah, so it's over 1,700 millimeter square, which is a lot of real estate for tissues.
Good
So it
point.
really goes beyond. And if you have any questions, like, because I think it's going to be also depending on the section and the sample that you have, so we can also help you make the calculations. As Egon mentioned, you can place multiple samples in one slide. The other questions, I mean, we have 5 minutes left, so it could be more about what type of training and support 10x provides. So there are two sides of this. There is the training that 10x provides to core facilities, but also the support that 10x provides to researchers that will be the customers from the core facilities.
Sure, yeah, great question. So when we set an instrument that comes with a on-site training, and I used to be a field application scientist, and I was a field application scientist at 10x for 4 years, approximately 4 years, and so every time we set an instrument, we provide on-site full on-site training. Having said that, that doesn't mean that you don't have additional support. So that doesn't mean that once the training is done, then you're not going to see us anymore. No. One thing that you always, you will never get with 10x is only one meeting or one phone call or one email. So that is really going back to my, last slide, what I was saying, we can really have outstanding resources to provide, to fully support you. I did mean that. So that is where, for instance, science and technology advisor like myself and Adriana will partner with customers from the very beginning of their experiment to help them design the experiment, choose the best approach, the best solution. Then we have FAS that will help them with sample prep, will help them with troubleshooting. And the same is true when it comes to supporting the core. Really, again, do not hesitate to reach out to us at any time for any type of question. We are really here to help you and make sure that you will be successful.
Awesome. And what one final question, I mean, we have 2 minutes, but what type of sample inputs do customers send to the core in general?
Oh yeah, that's a great question. So typically is a single cell suspension. So for single cell, typically is a single cell suspension or a single nucleus suspension. And then usually cores will take the sample, check the viability, concentration, then load the system, load the instrument, and make the libraries. With Visium, that depends. If a site assist is involved, there are cores that can, or certified service provider that can do the entire workflow, starting from so you submit them a tissue section on a glass slide, and they will do the entire workflow. Or it really depends. You might submit you might submit them or prepare a gene expression slide and then give it to the core. That's a little bit perhaps more or less standard, or it could be a little bit different depending on the cases.
Awesome, thank you so much, Egon. And I know there are some questions about still the throughput of Xenium, but we will make sure to follow up with you. I just want to leave you with this link that I just posted on the chat. So this is basically, it's also here. So it's basically the way you can connect with us and really continue these conversations. Thank you.
Thank you.
Once again, Egan, thank you very much. Thanks to the entire team from 10X to presenting this seminar. We appreciate it. As I mentioned earlier, we have another program next week on flow cytometry. You can find out those details on the ABRF website. We appreciate everyone joining us today. Look forward to seeing you once again online. Again, thanks everyone. Have a great day. Bye now.
Thank you.